The overall goal of this rapid method is to improve the yield of endogenous peptide hormones to be measured in human blood.
The rapid method was recently established for the use in rats and is also suited for the use in human blood. The rapid method improves the yield and allows the detection of the correct molecular form of endogenous peptides.
The rapid method is easy to use.
However, the rapid method is more time-consuming and more expensive compared to standard blood sampling, so it might be more applicable in the research setting. Visual demonstration of this method is critical, as the timely dilution of the samples as well as the elution are of great importance to ensure a proper peptide level.
Collect venous blood at a standardized time after an overnight fast from a forearm vein and process according to standard procedures or the rapid method. Instruct the subjects not to exercise or smoke before blood withdrawal. For standard processing, collect blood in chilled EDTA-containing tubes and centrifuge within 10 minutes at 3000 Gs for 10 minutes at 4 degrees Celsius. Collect the supernatant and keep at -80 degrees Celsius until further processing by radioimmunoassay. For rapid processing, immediately dilute blood 1 to 10 in ice-cold rapid buffer, pH 3.6, containing 0.1 molar ammonium acetate, 0.5 molar sodium chloride.